Okay, so right now we're starting part 2 of our enzyme lessons. So for part 2, the first thing I want to start with is a review of what we just talked about, which was the digestive enzymes. So the 3 key digestive enzymes that you need to know are amylase, which breaks down starches, protease, which breaks down proteins, and lipase, which breaks down lipids or fats. And amylase is going to be produced by the salivary glands and the pancreas. Proteases are going to be created by the pancreas and the stomach and the small intestine. And then the lipases are going to be produced by the pancreas and the small intestine. But in general, you don't need to know the locations, but I'm just giving you an idea of where this is. So again, as you take in a mouthful of food, what you're going to see, let me get this on here. Go back to full screen. So food will go in, and then right here, as it's first broken down, it's going to be broken down into disaccharides, but the proteins and everything will start there. So then it'll go down and into your stomach, and into the stomach, it gets pulverized into chyme. And this is again where pepsin, which is a protease, is going to mix with acid to help break down your proteins. The acid actually helps to unfold all the proteins. So then it goes into your small intestine, into specifically your duodenum, where then it mixes with bile, which bile breaks down your fats. And then the pancreas will inject more amylase, lipase, and protease to break everything down. Then in your small intestines, you'll have things called disaccaridases, which will break down all the disaccharides that are left over after the amylase breaks it down. And then finally, your last bit of lipases, and then you have a chemical called trypsin. So trypsin, it will function at high pH. So basically what happens is the small intestine will also have it will have things like the bases that basically neutralize out the acids. Okay, so that's a review of what we just talked about. On to the next part, homeostasis. So homeostasis is a condition where the internal environments of the body is stably maintained. Okay, and the purpose of digestion is to maintain the level of nutrients needed for the body to produce energy and to repair and grow new cells. So your body has set points for homeostasis, and they're based on maintaining optimal conditions. So the way you know you have a set point for something is like if your body changes to get yourself back to that set point. So for heat, you might sweat so that it can bring your body temperature back to the normal conditions. So as it says here, your temperature needs to stay near optimal, and your body will stay at 37 degrees or 90.6 degrees Fahrenheit. And this is hot enough for chemical reactions to take place, but not so warm that our enzymes lose their shape by melting. And we'll talk about what that means. So if you get too hot, you'll sweat, and if you get too cold, you'll shiver. So this is how your body basically reacts to different temperatures. So as it gets colder, what happens is molecules slow down. So the substrates and enzymes are not able to form the enzyme substrate complex, and therefore they can't interact as well. So you'll see a decrease in enzyme activity. But when you're at this optimal temperature, what you'll see is you'll have good shape to your enzyme, but also your molecules are moving around quicker. What happens as you get hotter is, although the molecules are now moving around quicker, what can happen is the bonds that hold the enzymes together, they start to break up, and that is called denaturing. Okay, so make sure you're taking a moment to write each of these different things. If you need to pause the video, you can. And also make sure you're adding the annotations to the graph that I have here. Your pH is also very important, and your pH is maintained by the combination of part of your brain called the hypothalamus, and then your liver and your lungs and your kidneys are all involved in maintaining your pH. So you basically need to maintain your blood at a slightly basic 7.3. But the thing you want to make sure that you understand is that 7 is neutral. Okay, 7 is neutral. And then anything higher than 7 is basic, and then anything lower than 7 is acid. So lower than 7 acid, higher than 7 basic. So you have some chemicals or some enzymes that are going to, most of them are going to work at your normal blood pH, which is about 7. Like I said, maybe a little bit above 7, like 7.3, 7.4. But you have some, like the ones in your stomach, that work at acid conditions, and then trypsin works at slightly basic conditions. And again, the point of that is the combination of that allows you to neutralize the food that are coming out of your stomach. So as this says, some can function at acid conditions, which is a pH of 0 to 6, like the ones in the stomach, and some can work under basic conditions, a pH of 8 to 14, like those in the upper small intestine. Now again, 7.1 to 7.9, those are going to be basic conditions too, and then 6.9 down to 6.1, those are going to be acid conditions. So you also need to have enough substrate. So like when you get hungry, your body tells you, hey, I need more food. And that's food are your substrates. So this, the substrates, this is your food. So when you need to eat or you need to drink, your body tells you that. So when your supply of needed chemical decrease, your body will respond. You'll breathe harder to get oxygen. You'll feel hungry to get more food. You'll get thirsty to get more drink. So some ways in which you do that, you are, getting stuff. And note, it says your body is able to run at less than 100% capacity. And this is what I mean by that. So I want you to draw in this graph. So you draw a graph that goes up and then levels off. And what you're showing here is that your body, in some cases, can run where you only have about 50% of what would be like a half tank of gas. So think of this as like the gas that you need. You can still run pretty much at full throttle on half a tank of gas. But then once your gas gets below half the tank, then you need to refill. You need to drink. You need to breathe harder. You need to eat more. Okay? So again, substrate's going to be your food, gases, and liquids that you need to survive. That's what we are talking about when we talk about substrates. Your enzymes, same deal. Your enzymes need to be at the optimal level. So your enzymes get made by your body in some cases. And one of the things that happens is some people have a lot of enzymes, and we say that they have high metabolism, and some people have low amounts of enzymes at the natural level. We say they have low metabolism. So their body needs more fuel to keep working, and some people, they need less fuel to keep working. So if you have a high metabolism, you probably need to eat a lot. If you have a low metabolism, you might need not to eat as much. Okay, so you could say like someone who has low metabolisms like here, whereas someone with high metabolism is like here. So they need to constantly be eating. But again, this graph of where it goes up and levels off, this is a key thing. And again, you should be seeing this over and over again. And we're going to look at this in lab conditions too. Finally, enzymes can be slowed or stopped. This is what drugs do in some cases. Toxins or drugs can act as fake substrates by binding to an active site, preventing the substrate from bonding. So one of the examples is caffeine. So there's a chemical that your body needs to respond to called adenosine. That's a stress hormone. And your body basically has an enzyme that breaks it down. Caffeine can block that, and by blocking it, it then will keep adenosine levels high, which basically keep you at a state of stress. And that's basically how caffeine keeps you active, because it's keeping you in a state of stress. Breaking down the enzyme, excess heat. So your body producing a fever, one of the things it's trying to do is trying to break down proteins. Preferably, it's trying to break down the proteins of like a virus or something like that. But you know, that still works the same way. It can do that on yours, which is why you have to kind of slow down your own metabolism by sleeping a lot when you do have a fever. So again, excessive heat or extreme pHs, it can cause the proteins to no longer function. So again, what we're drawing here is an actual enzyme and the actual substrate. But what happens with high heat or extreme pH, meaning like really low or really high, the enzyme gets unfolded, and now the enzyme, the substrate, can't make a contact. It doesn't have an active site. So the active site is gone. So denaturing can be reversed, but in some cases, once it's been denatured, it no longer will function. So like if it's been boiled, it probably won't function anymore. But if it's just been heated up a little bit, it might not work anymore. And then once it cools back down to the normal temperature, it then goes back to having its active site. So those are the key things about enzymes and what is needed to get the enzymes functioning, and how they can be stopped, slowed down, etc.